Hello and welcome to episode 10 of this series. And today's perfume is one of my favorites. It is Sycomore by Chanel. It's part of their special collection. These are large volumes. They're not that expensive considering how much you get, but they're still expensive. This is a beautiful fragrance composed by Christopher Sheldrake, and it's essentially a very fresh vetiver, which is ideal for today because it's a very hot day today. And when you put it on in the morning, you feel refreshed and clean. It's lovely. So in the last episode, episode 9, you remember that we talked about the fact that humans could tell isotopes apart provided you had enough hydrogens in the molecule. So that, for example, with acetophenone that only had 8 hydrogens, they couldn't really do it. And with musks that had 28 hydrogens, replaced by deuterium, they could do it really quite easily. So the question is, could we do this on animals? And there are 2 reasons for doing something on animals. First of all, animals cannot be influenced psychologically. So people feel rather more comfortable getting numerical results out of animal experiments where there's no possibility of suggestion somehow, or involuntarily hinting at something. And the other reason is that some animals clearly have a better sense of smell than we do. Notably, for example, dogs. As is well known, dogs can smell things quite well even inside a bag or from a distance, or through several feet of snow, or after several weeks and that kind of thing. So you might get lucky and find an animal that has a very good sense of smell. So when we were considering doing animal experiments, someone suggested I should work on fruit flies. So what is Drosophila? Well, Drosophila is called the fruit fly because you find it on fruit. It should really be called the vinegar fly because their favorite thing is vinegar. But you often find them in a glass of wine. If you're outside and you have a glass of wine on a table, you look in there and there are 3 or 4 Drosophila have fallen in and drowned. So what's special about Drosophila? Well, I didn't know much about Drosophila when I started, and I still don't. And I, in fact, ended up collaborating with a real expert on Drosophila genetics and behavior, my colleague Makis Skoulakis, who's shown here smiling. And the list on the right explains what is amazing about Drosophila. In fact, as I say, insanely great. The number one thing about Drosophila is that every single one of its genes has been sequenced, and it has 14,000 genes, that you can order from a place in Austria a strain of Drosophila, a particular line of Drosophila mutant, that has that particular gene turned off, and that's using a thing called an RNAi. We won't go into what that means, but basically you can order the strain with a gene turned off, and you get it in the mail, and it's cheap, and then you grow the flies, and it's also cheap. It has a short life cycle, which is useful for genetics. And many people, particularly in biomedicine, they say, well, Drosophila, there's just an insect. We're not insects. Yes, that's true. We're not insects. But 75% of the disease genes of Drosophila are the same or very close to the human ones or to vertebrate ones. So what this tells you is that, yes, evolution has made things look different. They put the skeleton on the outside. They made them small. They have wings and we don't, and so on and so forth. But at the same time, the basic machinery of life is pretty much the same in Drosophila as in invertebrates. And so they're extremely useful if you're interested in studying things that you cannot study either on humans or on mice because it's impossible or too expensive. The other thing about Drosophila, which is very unusual, is that it's a community of scientists that shares the results. So that if you create a new fly by some genetic engineering, a new strain of fly, by genetic engineering, you remove genes, you add this, you do that, you make it fluorescent, something or other. The moment you publish the research, you have to supply the strain that you've made to your colleagues. There's no exception. There's no exception. If you don't supply it to your colleagues if they ask for it, or supply it to a central library, then you basically are no longer part of the community, and it will cost you. So this is a very unusual situation, which is not, by the way, the case for things like mice, where if you spend a couple of years and a lot of money making a new transgenic mouse, nobody's going to expect you to share it with all your colleagues on day one. You keep it to yourself. So Drosophila is a very unusual community, and they all know each other. There's Drosophila people. I'm not really part of it. I just collaborate with Makis. Now, the other thing is Drosophila are not stupid. They learn, and they have a good sense of smell. And that's very important for our experiments. So how do you study smell in Drosophila? Well, like everything to do with Drosophila fruit flies, it's surprisingly cheap and easy. And in fact, it's rather delightful how direct the experiments are. So let me explain. If you want to know whether Drosophila prefers smell A over smell B, what you do is you put them in a what's called a T-maze. So you put them in a little chamber, and the chamber has access on both sides to two tubes into which you can put smell A and smell B. So in this case, I've put, let's say, deuterated and normal. And the flies are in the middle, and then they move in one direction or the other. And then at the end of a fixed period of time, you remove the tubes and count the flies inside them, which is very straightforward. And you write down the numbers and you start again. So the first thing we did was to ask whether Drosophila prefers a deuterium to hydrogen when it's in a molecule. So we started again with acetophenone. And the experiment basically consisted in putting the flies in the middle, as I've just described, and then on one side, no smell, and on the other side, acetophenone. And when you do this with normal acetophenone, and you look at how many percentage of excess flies there are on the acetophenone side, you find, that's the molecule of acetophenone, you find that with a normal, that is hydrogen acetophenone, the flies go towards the acetophenone. They prefer acetophenone to nothing. Now you can order deuterated acetophenone in several flavors, so to speak. One is with 3 hydrogens turned to deuterium, so that's D3 acetophenone. And when you do the experiment, you find that flies are still attracted somewhat less, but not statistically significant difference. They're still going towards the acetophenone D3. When you use D5 acetophenone, where the benzene ring has been deuterated, by contrast, you find that they avoid it, and that result is statistically significant. In this case, instead of going towards the acetophenone, they go away from it towards the nothing tube. And when you fully deuterate the acetophenone with sort of 8 deuteriums, that's D8, you find that a very clear result that they avoid the D8 acetophenone. Now, this is a very different answer to what we got with humans. As you recall, the humans could not tell, in fact, or not easily, it was very close, the 2 acetophenones apart. And so it suggests that flies have, in this particular respect, a better sense of smell than humans. It's also worth remembering that their receptors are completely different from those of humans. They have a completely different organization in the membrane. Instead of being arranged such that the carboxyl end of the protein and the amino end, these are the 2 ends of the protein, one up, one down, as in the vertebrate receptor, they're the other way around. So they still have 7 transmembrane helices, but they're different. So this is very interesting that they have evolved a different type of receptor, same number of helices in the membrane. There must be something about this magic number 7 of helices that does the job. Whatever mechanism they use, it looks like it can tell isotopes apart much better than we can. So I told you earlier that Drosophila was pretty clever, and you will see what I mean by that. You can train Drosophila to avoid an unpleasant stimulus. And how you do it is you put inside the tubes that contain the different smells, you put in one of the tubes, you put a track of copper that can deliver an electric shock to their feet. And when they go into that tube, they get an electric shock. And they don't like it. And they remember it, such that when you test them afterwards, they will avoid whatever smell was associated with that electric shock. And you can do this with deoctanol and octanol. With no shock, they have a slight preference for the normal octanol. With a shock on the octanol side, they go to the deoctanol, and that is statistically significant. And with a shock on the deoctanol side, they go to the octanol. So this shows you that Drosophila can actually learn to associate a smell with an electric shock. So now that we've established that the flies can A, tell deuterium from hydrogen apart, as in acetophenone, and B, can be taught to avoid one or the other, then we can start to play some really interesting tricks with the flies. And the first trick that we'd like to play on them, and the first question really we'd like to ask is this: does deuterium itself have a particular smell? In other words, does it behave like a functional group for flies? Is it like the way SH would be for us? Can they tell the presence of deuterium in any molecule? A clever experiment that you can do is to take flies that have not been trained against either a hydrogen or a deuterium molecule, and you expose them to, let's say in this case, benzaldehyde and dibenzaldehyde. So dibenzaldehyde on the left and benzaldehyde on the right. Deuterated on the left, normal on the right. And the flies, if the flies are completely naive, they haven't been trained against anything in particular, you find that they like them both equally, so they're pretty much in the middle. Now, if, however, the flies have been trained against octanol before, then they will avoid the normal benzaldehyde, the hydrogen benzaldehyde. Whereas if they've been trained against deoctanol, they will avoid the dibenzaldehyde. And that is truly remarkable. That means that they can smell hydrogen and deuterium in the octanol-deoctanol pair, and when presented with a different molecule, they decide that that molecule contains hydrogen or deuterium. So they're behaving as if for them, the hydrogen and deuterium behaves like a functional group in their smell. So far we've shown that they can smell isotopes, the difference between two isotopes, one molecule deuterated, one not deuterated, that they can be trained to avoid them, and that once they have been trained to avoid deuterium in one molecule, they will avoid it in another. However, this doesn't prove that it's vibrations. It could be some other weird property of deuterium that we don't know anything about. In fact, even though I can't come up with another explanation, some people just are not that convinced by these experiments. They want some more direct proof that it is vibration. And when we started doing these experiments, we realized that we could do a really clever experiment which involved vibrations directly. And it came from the realization that there happens to be a completely accidental overlap between two totally different vibrations. One is the CD stretch. In other words, when you replace hydrogen with deuterium, the stretch frequency of the carbon deuterium goes from, say, 3,000 wave numbers to about 2150, 2200. And the other one is the CN stretch, C triple bond N, which is at 2150, 2200 anyway. So it occurred to us that if the vibrational idea was correct, then when the flies were taught to avoid a deuterium, they would also avoid a C triple bond N, which is called a nitrile. And so this, if true, could not be explained by anything other than vibrations, because, of course, a CH and C triple bond N have chemically nothing in common. So this slide shows what the vibrations that I've just talked about might look like. So we have two molecules that smell lemony. One is citronellal and the other one is citronellal nitrile, and we've seen them before. One smells of lemongrass, the other one smells of metallic lemongrass because of the nitrile. If you look at the vibrational spectra, you find that the fingerprint region below 1500 on the left is rather similar, which kind of explains why they both smell lemony. But the big difference between the aldehyde and the nitrile is that in one case you have a big C double bond O stretch of the aldehyde and also a strange aldehyde CH stretch. And in the other case you don't have an aldehyde group at all, so those two things respectively at 1750 and 2800 or so are gone. And instead you have a small peak at 22 something, which is in particular cases seems a little bit calculated, a little bit high, but nevertheless it is in that region. So in the previous experiments we've shown that the flies could cross-learn from deuterium to deuterium. In other words, that once they had been taught to avoid a deuterated compound or a normal compound, they would avoid the normal or deuterated compound of another pair, in that case benzaldehyde. Now what we're doing now is more striking, which is we expose them to, on one side, the lemongrass nitrile, and on the other side, the lemongrass aldehyde. And when they haven't been exposed to anything at all before, they find them equally attractive or equally repulsive. It's hard to tell in this particular experiment. And they are quite similar. In fact, the nitrile is used as a replacement for the aldehyde in many household products where you want the lemon smell, but you want it to be stable in bleach, because nitrile is stable in bleach. Now, when they are trained against the normal octanol, nothing much happens. And I'll explain that one in a minute. But when they're trained against the D-octanol, then they avoid the nitrile. And that really is remarkable. They avoid the nitrile only if they're trained against a molecule that has the same vibration as the nitrile. So you may ask, how come the flies did not respond in any way to the normal octanol? Well, we had a lot of trouble in the beginning understanding that result. It seemed kind of weird and counterintuitive, but actually it turns out to be very simple. If you shock the flies against octanol, which has hydrogen atoms, CHs, and you ask them, smell these two, citronellal nitrile and citronellal, well, they both have CHs. They're the same from that standpoint, and so the flies are indifferent. So it makes perfect sense. So when we figured that one out, we were very relieved because all the results made sense. When you shock them against a CD vibration, then they look at the two molecules and they say, oh, the nitrile is kind of like CD, I'll avoid that guy. When you shock them against a CH molecule, they look at the two molecules and they say, both molecules have H's in there. I don't know what to choose. I'll choose nothing. Well, they're equally bad or good to me. So in the end, it turned out that this weird result fit perfectly in the general scheme, which is that they smell vibrations.